Good morning, students. In the last class, we had discussed about the pollen-pistil interaction. Okay, on the basis of chemical components which are present in the pistil and pollen grain and the stigma or the pistil. There is an interaction between this pistil and pollen grain. So that is the thing. The pistil will accept or either it is going to reject the pollen grain. So if it is of right type or compatible, at that condition, the pistil will accept the pollen grain. Okay. If it is of wrong type, at that time it is going to reject. That we are calling it as the incompatible pollen grains. Okay. So in that things, we also studied how the pollen grain is going to germinate and how the pollen tube is going to grow. Okay. The pollen tube will enter through the micropylar region of the ovule. Okay, when it is entering through the micropylar region of the ovule, in the micropylar region there will be presence of two synergids. That synergids are with filiform apparatus. That filiform apparatus is guiding the pollen tube to enter into the ovule, embryo sac, where they will go or passes, then it is going to fuse with the cell. So that all things we had studied. So here next topic in this chapter, that is the artificial hybridization. So let's see about that one. Artificial hybridization. See, in the chapter strategies for enhancement in food production, already you had studied what you mean by artificial hybridization. That is, in animals or in the plants, the two animals or the two plants are making artificially to mate them. Okay, due to which a new hybrid, that two parents, that is male and female, either they belong to the same species or either they belong to the different species. Okay, so that is the thing a hybrid is going to produce, and that hybrid we can say, which is that hybrid will have the characters of male of one species and characters of female of another species. See, in animals you had studied in artificial hybridization or hybridization, that is what we call it. It may take place among the same species or among the different species. One example I will tell you, that is what we call interspecific hybridization. One example, mating with male horse mating with a female donkey. Okay, so both the characters you can see in a new hybrid that is named as the mule. Okay, so like that only here also. Why this artificial hybridization is going to take place means to improve the crop program or crop improvement program. It is one of the major approach. It is one of the major approach to crop improvement program. Okay, here why the artificial hybridization is practicing it to improve the crop management or these programs which are associated with the crop or agricultural field. So in such crossing experiment, it is important to make sure that the pollen grain should have the desired characters. Okay, so here the pollen grains, whichever they are going to use, it should be with desired characters. Means, for example, this is the plant, one plant. This is the flower, and this is the male flower, anther. Okay, stem and what we call. So here, what they are doing artificially, not naturally. Okay, This is one plant, and what is in this plant? What is the desirable character means? That is high yield. It gives a higher amount of crop or whatever the food you want from the plant. Okay, one more plant. This is the female parent. So here you can see this is the stigma. Okay, so here what is the desirable character over here means that it is a what we call that is disease resistance. Okay, so what you want in the next generation when you cross both of them? Okay, cross means we are going to mate them. How means we are taking the pollen grain from this anther and we are going to dust on it on this stigma. Okay, not naturally. Here there is no involvement of pollinators or pollination agents. But we are, or the researchers or the scientists, they are going to take the pollen grain from here by using forceps and all. I will explain you later. And they are going to dust on the particular stigma. They have the knowledge that this female parent has such a desirable character, and this pollen grain, whatever they are going to release dusting or taking away from the anther, it has the desirable character. What is that? High yield. They have all that knowledge. By having that knowledge, what they are going to do? They are making them to cross. So whatever a new hybrid or whatever a new offspring is going to produce, so that we can call it as the hybrid. So this hybrid will have both the characters, that is the disease resistance and high yield. Okay, so this is what somewhat characters are going to improve over here. For what reason artificial hybridization is practicing means to improve the crop improvement program or to improve the crop characters. Okay. So here and here, first of all, the pollen grains should be taken out. It should not be a size when anther gets mature. Whatever the breeders will be there, or plant breeders, or whoever, or a person who is having the such type of knowledge, they know the time when anther is going to mature. At that time, they will take away all the pollen grains by using a forceps. Then they are going to dust it on the stigma. Okay, so in this way they are going to practice the artificial hybridization. So here, these particular steps, whatever removing the anther, pollen grains and dusting on the stigma, it should not contaminate with some other pollen grain. We want only this character, only this pollen grain. So it should not contaminate or no any other pollen grain should come and sit on this stigma. Okay, so these all things is achieved. And the artificial hybridization can be achieved by two methods, that is emasculation and bagging method. Let's see about that one. This artificial hybridization can be achieved by emasculation and bagging technique. Which are the two methods by which we can practice this artificial hybridization, that it is the emasculation and bagging technique. So let's see about this emasculation and bagging technique. So first up, we will see about this emasculation. Emasculation and hybridization. See here one female parent is there. This is the plant and this is the female parent. That means female flowering plant. Okay, which has only the gynoecium, not androecium. So this female parent produces bisexual flowers. Okay, this is the bisexual flower. Bisexual flower means that it has the stigma, pistil, then the stem. Bisexual flower. This is the pistil and this is the stem. Okay, so here what happens means when flower is not bloomed or when this flower is at bud stage. Okay, when the flower is not open, it's still bud. What they will do means, so here they are going to remove the anthers from that particular bud. What they are going to do? They are going to remove. See, for example, this is the bud, and here I will show you. This is the bud in which there is a presence of pistil and the anthers. Okay, so what they are going to do is they are going to remove these anthers from this bud. Artificially, they are going to open it. Okay, then they are going to remove all these anthers from this bud. See here, by using the forceps, they are going to cut it or they are going to remove it. All the anthers have been removed from this bud. Now there is no anthers. Okay, in this sense, artificially they are going to remove the anthers from this bud and anther when see, before dehiscing means before maturation of anther, they are going to remove the anther from the bud. If flower is going to open, at that time only the anthers are going to mature. They are going to remove it at the bud stage. That means the anthers are not matured. Okay, before dehiscing the anther, they are going to remove all the anthers from the bud. So what happened means this step is the step where there is a removal of anthers before they get dehisced. Removal of anther before they get dehisced by forceps. By using the forceps, this step of removal of anther by forceps from bud. Okay, this step is known as the emasculation. What it is known as? It is known as the emasculation. So if see what is happening if anthers are removed from the bud means when flower is going to open, at that time here there will be no self-pollination is going to take place. Okay, self-pollination means self-pollination results in the inbreeding depression. That characters are only going to pass to the next generation, but we are undergoing into the artificial hybridization. We want some other plants characters. Okay, so that is why we are going to remove the anthers. Okay, the next what happens means now the emasculation flower or this is the emasculated flower. Emasculated means from where the anthers are removed. Now this flower we can say it as the emasculated flower. So what happens means this flower is going to tied or covered with a paper, that is the butter paper or a polythene bag. Here you see this flower is covered with a butter paper or a polythene bag. See, emasculated flower is covered with butter paper or polythene bag. And this step of covering or closing that particle, why they are going to cover with so that no any other pollen grain, undesirable pollen grain should not contaminate this flower or the stigma. Okay, so this step we can call it as the bagging. Covering the emasculated flower with butter paper or the polythene bag, so that no any other pollinators or pollinating agent should come and disperse the pollen grains of some other plant. Okay, this step is known as the bagging, where the emasculated flower is covered with polythene bag or butter paper, so that there should not the stigma should not contaminate with the unwanted pollen grains. Okay, the next when the stigma of bagged flower attains receptivity, see still this flower is at bud stage, still the stigma is not matured. Okay, we had removed the anthers before maturing only. Now, what happens means till what time they will keep this cover lips till the stigma won't matures, or it should the stigma should mature, and it should mature at what time means it should become able to receive the pollen grains. Okay, till that time they are going to bag. So next thing, now next whatever the pollen grain whatever the pollen grain with the desirable characters will be there. So at that time, what they will do means, see here, this is the male flower with the stigma. Another flower or the desired pollen grains or desired stamen. Okay. So here, this is the flower. What they are going to do means, they are going to remove the anthers from the stigma. Then they are going to, and these anthers are matured anthers. Okay, what they will do? They will take one petri dish, and they are going to dust it on this petri dish, so that all the pollen grains should disperse in this petri dish. Otherwise, what they will do? Directly they will take these anthers, okay, or these stamens, and they are going to remove this cover or this bag. Okay, now this flower is matured, so this flower is opened now. Okay, stigma also matured. So this is the stigma. Now they are going to remove only for a certain time. Then they are going to take these anthers, then they are going to dust. This is the anther with desirable pollen grains. They are going to take near to the stigma, then they are going to dust it on the stigma. Okay, what they are going to do? They are going to dust it on the stigma, so that these pollen grains are desired pollen grains. They want these pollen grains. Then again, they will remove this, they will take away this anther. Again they are going to rebag this flower, so that it should not get contaminated with some other pollen grain. So next, what happens? The post-pollination events are going to take place. The fertilization will take place, seed development is also going to take place, then the fruit development is also going to take place. Okay, so in this step or this method, which you can call the removal of anther, that is the emasculation, and covering the flower with the polythene bag or the butter paper, that is known as the bagging technique. The next term, these all things we are talking about if the female parent is a bisexual, or the female parent plant is having the bisexual flower. In that condition, all these things are going to take place. If the female parent bears unisexual flower, means what happens? Okay, let's see here. If a female parent produces, this is the female parent, female parent produces unisexual flower. Unisexual flowers means only the female flower or the male flower. Any of one of them, not both. Unisexual means only one sex organ or one reproductive structure will be there. So there is no need of emasculation. Whatever you are going to remove the anther, that is no need, because there is a chance of self pollination, but here there is a cross pollination can take place. So no need of emasculation. When the female parent bears the unisexual flower, there is no need of emasculation. Okay, this is one thing. And the female flower bud, see what they will do in the unisexual flower means whenever the female flower bud, at that stage only, when the female flower, this is the female flower in which there is the presence of stigma. When it will be in the bud stage, this is the bud stage. So what they will do means they will cover, or bagging technique is going to employ. They are going to cover it with what? With the butter paper or the polythene bag, so unwanted pollen should not come and sit on this flower. So when stigma matured, or when flower gets matured or flower is going to open, at that time, for example, this is the flower is opened now, or the flower becomes matured and stigma also matured. At that time, what they will do, they are going to dust the desired pollen grains. By removing this cover, they are going to dust the desired pollen grains. Then again it is going to cover with the polythene bag. In this way, the post-pollination events are going to take place. Then this will give up this process. We can say it as the artificial hybridization. Okay. The next topic in this chapter, that is the double fertilization. So let's see about that one. Next topic, that is the double fertilization. See here, this is very important question for 3 marks in your theory. So here, see, during pollen pistil interaction, what you had studied with the see, during pollen pistil interaction, what happens means one male gamete. Here I will show you. This is the amphiocyte. This is one. This one, where two synergids are there. One egg cell is there. Okay. Within these two synergids, a filiform apparatus is there. This is the filiform apparatus. Okay. So here, what happens when the pollen tube enters through the micropylar region? It has the two male gametes. These are the two male gametes. This pollen tube releases these male gametes into the synergids. Okay. So, for example, this is the synergid. This is the what we call nucleus of the synergid, and here this is the male gamete. The male gamete I will represent it with the green color. So in this way, they are going to release the male gametes into the synergids. Then what happens means one of the male gamete gets fused with the egg cell. Here I will show. This is the egg cell. Okay, and this is the one male gamete. Okay, see here. This is one egg cell, and this is the male gamete. One male gamete fuses with the one egg cell. Okay, and in this condition, here the egg cell is haploid. The egg cell is haploid, and the male gamete is also haploid. Okay, haploid plus haploid, it will become diploid. Okay, here the egg cell fuses with one male gamete. They both are haploid. Fusion of one egg cell with the what we call with the haploid male gamete. Okay, fusion of or else you can also write syngamy, fusion of male gamete with the female gamete. Okay, here I will write fusion of male gamete with the female gamete, similar to the fertilization. This fusion of male gamete, here it is the male gamete with egg cell, which is the female gamete. They both are going to fuse to form a diploid zygote. This process is known as the syngamy. This is the first fertilization which is taking place during fertilization. Okay, understood? The next one more fertilization is going to take place. What about the other cells in this embryo sac? Now comes the next cell, that it is the central cell or the polar nuclei. First, it gets fused. One cell is going to fuse with the egg cell. One male gamete is remaining. So that one remaining male gamete, it is going to fuse with what? Still here, there is an presence of a central cell as well as there is an presence of antipodal cells. So let's see what happens over here. This is the syngamy. See here, if they are asked for 3 marks, this process if you explain, you will get 1.5 marks. Only syngamy. Then one more is there, that is the triple fusion. Now I am going to explain that will carry 1.5 marks. Okay, it all includes the 3 marks. So see here, second one. What is the resultant cell of the syngamy? That is the zygote, diploid zygote. So here next term, what happens? This one male gamete, one is fused with the egg cell, which is formed the zygote, diploid zygote. Remaining one egg cell, it will fuses with the central cell or the polar nuclei. This one is the haploid male gamete, and these two are, we can call it as the central cell or polar nuclei. Okay, so here already the central cell will have the 2 nuclei. Okay, so what you had studied in 8 nucleate 7 celled stage. Okay, so already it will be having 2 nuclei, haploid, haploid. Okay, that is the diploid. Already they are haploid, haploid. Already they are diploid. That means central cell or polar nuclei is already diploid cell with the 2 nucleus. One egg cell, sorry, one male gamete is going to fuse with the central cell. What happens means it is going to form a triploid cell. And that triploid cell here we can call it as the PEN. What is the full form of this PEN means primary endosperm nucleus. What is primary endosperm nucleus? Okay, here you see, once again I will tell you, central cell already which is diploid because there is a fusion of 2 cells. Fusion of only nucleus, not cell. Okay, central cell plus the 1 male gamete, and this male gamete is the haploid. Okay, see here, this process we can call it as, here the fusion of 3 cells are going to take place. That's why we are calling this process second one, that is the triple fusion. What it is? It is the triple fusion. Okay, see here, central cell or polar nuclei, they will have the 2 nucleus but 1 cell. 2 two nucleus means it will be diploid. Okay, then one male gamete is going to fuse with the central cell. It will form a triploid cell. What is that triploid cell? Means that is the primary endosperm nucleus. Okay, so it will be triploid. This primary endosperm nucleus, it will be triploid, and there is a fusion of triple nucleus of the three nucleus. That's why we had named this process as the triple fusion. So this is the second fertilization in the flowering plants. That is the thing we have named it as the double fertilization. Two times the fertilization is going to take place. One is going to form a zygote. Next fertilization is going to form a primary endosperm nucleus. And here, what about these antipodal cells? These antipodal cells, these three antipodal cells, these cells will degenerate. Okay, these antipodal cells are going to degenerate. So, since two types of fusions are going to take place, that is one is syngamy and the another is the triple fusion. So, these together we can, which is taking place in the embryo sac, and this phenomenon is known as the double fertilization. See here. Syngamy and triple fusion taking place in embryo sac. This phenomenon is known as the double fertilization. Okay, so altogether this we can call it as the double fertilization. Next here, which is taking place in what? In the flowering plants, in flowers, in bare leaves, in the embryo sac, this double fertilization is going to take place. Okay, next thing, after the formation of zygote and after the formation of primary endosperm nucleus, further, after post-fertilization event, after fertilization, what happens means this primary endosperm nucleus develops into endosperm. What happens? This primary endosperm nucleus develops into what? Endosperm. This is very important point. Primary endosperm nucleus develops into the endosperm, and here I will write zygote will develop into embryo. First, second, zygote will develop into embryo. Okay, so these are the two things after double fertilization. What is going to take place means there is a formation of endosperm and the embryo. Zygote will develop into embryo. Primary endosperm nucleus will develop into the endosperm. Keep in mind, zygote develops into embryo. Primary endosperm nucleus develops into the endosperm. Okay, fusion of one male gamete with one egg cell. That is this process is known as the syngamy, which results in the formation of zygote. Zygote further it will develop into embryo. Okay, then fusion of one male gamete with polar nuclei. It will form a triploid cell, that is the primary endosperm nucleus. further which is going to develop into the endosperm, and this process is known as the triple fusion. Okay, so this is about the double fertilization. Next we will see about the post fertilization events and their structure in the next class. Okay, thank you.